Hello everyone and welcome to Pocket Size Bio of Dr. Casco. In today's video, we're going to continue Chapter 7, Membrane Structure and Function. We'll be going through 7.3, which is passive transport is diffusion of a substance across a membrane with no energy investment. Also 7.4 and 7.5. So let's get to it. Okay. Okay, 7.3. Passive transport is diffusion of a substance across a membrane with no energy investment. Okay? As I mentioned before, when we talked about thermal energy in molecules in chapter 3, molecules have thermal energy, meaning that they're in constant motion. So if they're constant motion, they're constantly moving, and because of this constant movement, you get something called diffusion. Okay? Diffusion is the movement of particles of any substance that ultimately, enables these particles to spread out in the available space that they're in. And it could be directional. Okay? If you're in a certain area within a room, if you spread, a perfume in a concentrated area, these particles of the perfume will eventually spread out. Okay? And ultimately, the movement of these particles leads to something called dynamic equilibrium, where the particles, they even out within the space. So they're spread out evenly. There's no high concentration area as opposed to another area that has less. Although it might start like that, eventually the particles will spread out. So it's the movement of particles to eventually spread out through a space. Okay, if you see here, you have this figure 7.11. And let's say you have a synthetic membrane in some, a container full of water or whatnot, and you have an initial concentrated area of a dye. And this membrane has little holes in which the particles can move through. The particles will eventually, just through random motion, will spread out, cross through the membrane until you have an even movement. They're still moving around, but there's just a net movement where one area has a similar amount of particles as the other area. You would refer to that as being in dynamic equilibrium. Dynamic meaning that they're always in motion, but nevertheless, as one particle is going to one direction, the other one is going in the other direction. So there's a net equilibrium there. Okay, a little bit of rules here. In the absence of any other force, a substance will diffuse from where it is more concentrated to where it is less concentrated. Okay, in other words, a substance will move down a concentration gradient. So this is a very important term, concept to know. Biology is filled of examples where you have concentration gradients. So make sure you understand this term. And it's the same thing. You have an area of high concentration. You have an area of low concentration. That difference, okay, is referred to as a concentration gradient. Okay. And substances will generally diffuse from an area of high concentration to an area of low concentration. That in itself is diffusion. Okay. As I mentioned before, this is random motion. So diffusion is spontaneous. It is due to the just molecular motion of the particles. So no energy is needed. Okay. There's a movement in which one particle will move on its own gradient, high concentration, and then to low concentration, independently of any other particle. Okay, so if you see here, you have two different dyes. Okay, and you have an orange dye, you have a purple dye. Of course, each has their own area of high concentration. The movement of the orange particle will not interfere with the movement of the purple dies. Okay, so until they're in both in equilibrium, independent of each other. Okay. So when a substance has a higher concentration on one side of the membrane, it tends to diffuse through its own concentration gradient. Of course, this does come with its own little caveats. If it is permeable, if it can pass through, um, uh, if it can pass through its the membrane. Okay, sorry about that. Okay, just to give you an example here, oxygen will diffuse into the cells as long as they are undergoing cell respiration. Remember, as oxygen goes in through the bloodstream and into the cells, if the cells are consuming oxygen, it means that inside the cells there's always a low concentration gradient. So as blood is coming, bringing oxygen rich blood, it will diffuse into the cells. Okay, oxygen consumption, cell respiration. We're going to see this in a lot more detail also with gas exchange. All right. The diffusion of a substance across a biological membrane is called passive transport. So this is a term when you're talking about diffusion, passing through a cell membrane, it is referred to as passive transport. So no energy is needed. Make sure you understand that no energy is needed to make the molecules move from one place to another other than their own kinetic energy. Okay? When you have, we're going to see this a little bit later, a concentration gradient, that in itself is a form of potential energy, and it could help in ATP production. Okay, and we're going to see that when we get into cell respiration and photosynthesis. Okay, let's continue on. A special type of diffusion is referred to as osmosis. You probably have heard of this term before. Okay. And this is a special type of diffusion relating to water and water balance. Okay, so water diffuses across a membrane from a region of high free water concentration. So that means that you just have a um, a beaker, full of just water where there's a relatively low solute concentration. Okay, to that of a lower free water concentration. Okay, or an area where there's a high solute concentration. Okay. Ultimately, just to give you a general sense, water likes to go from where there is less water. Okay, free water concentration likes to go to where there's less water. Ultimately, until the solute concentrations are equal. Now, there's a particularity with this, right? The diffusion of water occurs through a selectively permeable membrane. Okay, this is called osmosis. Now, if you take a look here, let's say you have the system here. You have a glass tube, and in the middle of this glass tube, you have a permeable membrane. And on either side of this membrane, you have sugar, but on one side you have a low concentration of sugar, on another side you have a high concentration of sugar. And then if you were to add equal volumes of water to each side, or just enable them to fill up, just because one area has less water concentration relative to the solute, water will diffuse to the other side. Okay? To enable an equal balance of water until you have ultimately a good balance. Okay? Now, this unequal columns happens because sugar cannot pass through this permeable membrane. They're too big. Okay? So only water would pass. So this is what osmosis is referring to, the movement of water until you get a balanced concentration of water and ultimately solutes also. Okay. And this behavior, this phenomenon has special effects on cells, right? Animal cells and plant cells. So we're going to take a look at what happens with osmosis in organisms that don't have cell walls. Okay, so some concepts that we need is, of course, solute concentration that we've talked about already. Um, how much you have of whatever you want to dissolve inside the solvent, in this case, water. Okay, and permeability, the ability for something for the cell membrane to enable things to flow in and out. Related to this, and this is more specific when you're talking about osmosis, is tonicity, the ability of the surrounding solution to cause a cell, okay, to gain or lose water. Okay, it's called tonicity. Now, again, here, the surrounding solution to cause a cell. So we're referring to a cell that is submerged inside an aqueous solution. Okay. Now, this depends if a cell is going to lose water or not, or gain water, depends on how much solute is in the solution and if they can pass through the membrane. Okay. So let's take a look more specifically at this. So in the case of cells without a cell wall, let's say animal cells, let's say more specifically a red blood cell, you can have several situations. The first situation, where let's say you get a red blood cell and you submerge it in a solution where the solute concentration is similar to the inner cellular content of solutes, you refer to this as being an isotonic solution or an isotonic environment. So there's no net movement of water because water is diffusing at the same rate as water's coming into the cell, water's leaving the cell. So the cell volume is relatively stable. And this is due to the environment being in a similar solute concentration as the internal contents of the cell. So it's isotonic, stable. Okay. You can have another environment where you have a hypertonic solution. This is high solutes within the environment. So because of this, remember the solute concentration outside of the cell is incredibly high. Water likes to go where there's less water or where there's higher solute concentrations. So water will leave the cell, okay, if it does not have a way to thrive in this solution, high solution, solute solution, it will die. Okay, it will shrivel, dehydrate completely, and ultimately die. And this happens when you have this can happen in areas with high salinity, lakes, right? They could kill animals if they don't have a way to be able to moderate a water loss. Hypertonic solution. In this case, for a red blood cell, it will shrivel. You have a hypotonic solution. This is referred to as a low solute environment where water will enter the cell faster than what it can leave. Okay. Remember, water likes to go where there's less water. In this case, you have a more solute inside the cell, so water will go inside the cell, okay, because there's less water. All right, so water will enter the cell faster than it leaves. Ultimately, in the case of red blood cells, it could eventually swell to the point that it can't not withstand this pressure and it bursts. Okay, this is called hemolysis. So you have isotonic, hypertonic solutions, hypotonic solutions. Okay. Seawater is generally isotonic to many marine invertebrates because they've evolved to withstand this environment. And land animals were protected from the environment because we have extracellular fluid that is a similar solute concentration, right, inside relative to the inside of the cell. So it's isotonic. So we have a way to kind of protect ourselves. Okay. Organisms without cell walls need to have a way to withstand fluctuations. Okay. against osmoregulation. So we need some form of regulations. Of course, we do. Okay. That idea of changes in salt concentrations and being able to control these changes and acclimate to these changes is referred to as osmoregulation. Okay. The control of solute concentration and water balance. We'll see this in a lot more detail also. Osmoregulation. Okay. An example of osmoregulation is the paramecium caudatum. Okay. You have this little protist here. And they have specialized by having contractile vacuoles. So these are single-celled organisms. They live in fresh water, pond water. So that this environment is generally hypotonic to the cell. So the plasma membrane, yes, it is generally less permeable to water. Water uptake does occur relatively slowly, but still it enters continuously into the cell. So the paramecium needs a way to release excess water. If it doesn't, it will burst. Because it has a contractile vacuole, this enables the paramecium to squeeze out excess water. It's a specialized structure that can enable water to pump out of the body. Therefore, the paramecium can thrive in fresh water. Okay, and these little things they're constantly contracting. Okay. Bacteria and archaea have similar adaptations to release water content. Okay, let's continue on. I will be using the same panel here for plant cells. Now, in situations where you have water balance with cells containing cell walls. Let's take a look here. So cell walls, you have organisms like plants, prokaryotes, fungi, some protists can have these thick structures. They undergo a similar situation: hypertonic, hypotonic, isotonic solutions. Okay. In the case of hypotonic solutions, okay, the cell wall helps maintain the structure of a cell, okay, in water balance. And remember, hypotonic, meaning water is coming inside of the cell. So let's just zoom into this really quick. Okay. So water is coming inside each individual cell wall, each individual cell of the plant. Okay. But because of the cell wall exerts a pressure against this incoming water. So it can help balance and maintain itself pretty well. Okay. That back pressure on the cell due to the cell wall is referred to as turgor pressure, opposing further water uptake. So in this sense, plants can sustain a high amount of pressure relative to animal cells. You refer to cells that are in this state, that are nice and fluffy, they're referred to as being turgid. They're in a good, healthy state. And a lot of herbaceous plants are in this state. You want plants to be like this. But let's say you enter a hypertonic solution. Cells can become flaccid. What happens here? Water is leaving the cell, so much so that the plasma membrane can peel off from the cell wall, okay? And as water is leaving, the cell membrane and the whole cell itself will kind of begin to shrivel. It's drying out. You can see certain sections of the cell membrane still trying to attach to the cell wall. And at this point, you say that the cell has plasmolyzed. Okay, plasmolyzed. It is possible to kind of regain cell back, the water back, and make the plasma membrane fill up again, and the cell fill up again with water, so it could get plumpy again. But ultimately, this occurs whenever the plant begins to wilt. Ultimately, if you leave this the plant wilted for too long, they'll eventually die. Okay. So the cell wall in this particular situation has no advantage. It can't help plasmolysis, prevent plasmolysis. Okay. So cells lose water to the surrounding and shrink. Okay, that is called plasmolysis. Bacteria can also exhibit plasmolysis. Okay. Okay. Going more specifically now, so we've talked about diffusion, we've talked about osmosis, movement of particles. Now, let's talk about facilitated diffusion. So now we're getting into the cell membrane particular, and we're referring to this particular location of the cell membrane. Okay. Passive transport that is aided by proteins that are embedded in the cell membrane. Okay? So the way that you have this idea of diffusion working is referred to as facilitated diffusion by the help of proteins. So you must have proteins in order for it to be considered facilitated diffusion. Okay, and of course the membrane. And it's very, very specific. So as I mentioned before, proteins, they have binding sites, they have binding areas that are very particular to a one specific molecule. Okay. So there's two types of facilitated diffusion. Remember, it's a passive transport, so there's still no energy involved. Okay. You have channel proteins and you have carrier proteins. Channel proteins, as you can see here in the figure in the diagram, it provides a channel, a tunnel, a corridor that can enable a particular solute to move across the membrane. Okay, these are generally hydrophilic pathways that will enable hydrophilic and polar molecules to generally move through the membrane relatively easily. Okay, at a good rate. Okay, at a higher rate. A very famous example is aquaporins. Okay, aquaporins. It's good to memorize this particular protein. It's a very go-to molecule to think about when you're talking about channel proteins, and it's just the movement of water. Very, very useful for plant cells. Even in our bodies, we have a lot of aquaporins and kidney cells because we want to reabsorb as much water as we can, okay, and prevent loss of water in our bodies. Okay, there's an example. Oh, I don't have the diagram of aquaporins. I'll check that in a little bit. So we have aquaporins. Okay, but ideally, essentially, you have, let's say, little water molecules, and they're consistently and constantly passing through these protein channels. Okay, and this could go vice versa, depending on the concentration gradient. Remember, it is passive transport. You have special types of channel proteins that can transport ions. They're called ion channels. And they can function as gated channels. We're going to see this a lot more in detail in a little bit. So these gated channels can open and close due to a stimulus. Okay. And the stimulus could be electrical, an electrical impulse. You see this more for nerve cells. An example of this is a potassium ion channel protein. Okay, you can see it's from an overhead view. You can see this little pore and opening there where potassium ions can pass through. Or it could be chemical. The channel can open and close based on some signal molecule that can bind to the protein, enable the protein to open up or close, depending on the situation there. Okay, so channels. Carrier proteins, these are glucose carrier proteins. They're specific proteins that enable specific molecules to pass through the membrane where the molecule that binds to the protein will attach to it. An example of this is a glucose transporter. So if you see here, you have this particular carrier protein. It has a binding area that is specified for a particular thing. In this case, let's say glucose. Glucose comes in. When it binds to the protein will change shape and enable the solute glucose to exit into the other side, inside the cell, or vice versa outside the cell, depending on the concentration gradient. Okay, so it helps translocate. It's a key word for you to use. Okay, it translocates any solute by changing its shape. Okay, across the membrane. Again, no energy is needed depending on the movement within the concentration gradient. Okay, let's continue on here. 7.4 now, we're going to talk about active transport. So active transport in itself uses energy to move solutes against their gradients. Whenever a cell wants to create a concentration gradient, it needs to pump solutes across the membrane, and those solutes need to go already where there is a crowd or a high concentration of that particular substance. So it goes against the gradient, and that requires work. This process, which that occurs, is called active transport. And of course, the name tells you it requires energy. Okay, so we're going to talk about active transport now. This type of movement of substances, of materials, requires carrier proteins. Okay, here you can see a summary of what I've talked about in the previous sections, right? Passive transport and diffusion in general, facilitated diffusion of ion channels or channel proteins and carrier proteins, enabling facilitated diffusion. And now we're going to talk about active transport here, and they're moreover a form of pumps, right? That you're forcing materials into a gradient. And yes, the cells do require to have constant gradients for some particular substances that we're going to take a look at. So this process enables a cell to maintain different concentrations of materials internally, right, the cell's internal contents from the extracellular environment. An example of this is whenever, and we're going to see this a little bit later, is when you want to have a concentration gradient of potassium ions on one side of the cell and sodium ions on the other side of the cell, relative to the other surroundings. Okay, so the way that a cell membrane does this or enables the cell to do this is by using protein, particular carrier proteins that are more specifically called protein pumps. Okay, moving potassium into the cell and sodium out of the cell. You always require energy, so there's going to be a hydrolysis of ATP supplying that energy onto the carrier proteins, as you see here, and we're going to see in more detail. And then that third phosphate will bind directly to the protein, enabling the protein to change shape and basically transfer any particle that is required, any ion, from one area to the other area. A very famous example is the sodium-potassium pump. Okay, so this is a general schematic of this whole process of sodium-potassium pumps, right? But we're going to see this in a little bit. So I'll get back to this drawing, to this diagram here. But in a nutshell, you can see, you have this particular pump where sodium is a high concentration in the extracellular fluid. Potassium is in a high concentration inside in the cytoplasmic area of the cell. And then this pump will enable sodiums to continuously get into this structure, sending them outward with the use of energy and that phosphate that you see there. Okay. Again, I'm going to go into a little bit more detail later on. So one of the things that is necessary for cells to function, and especially with nerve cells, is the idea of having membrane potentials and voltages. Okay, electric potential energy. All cells have voltages across their plasma membranes. Okay, voltage is a form of electrical potential energy, right? And It's created from this separation of opposite charges. So you need to have differences in ions to create some electrical difference, okay, uh, throughout the cell membrane. The cytoplasmic side of a membrane is generally negative relative to the extracellular side. So you would have a more positive side on the extracellular side just because you're more negative on the inside. So this is done or carried out because there's an unequal distribution of anions, okay, and cations on both sides. So when you're referring to voltage across a membrane, it is called membrane potential, okay? And depending on the cell type and depending amount of anions and that you have, this the measure of voltage can range from 50 millivolts to 200 to negative 50 millivolts to negative 200 millivolts. Okay, and the negative sign within these measures indicates the inside of the cell is negative relative to the outside. Okay, so have that in mind there. Internal environment is more negative. So membrane potential and having that difference acts like a battery. So it provides an energy source, essentially having a positive and negative side, right? Providing an energy source that affects all traffic, okay, traffic for all charged substances across the membrane. The inside of the cell is negative. So let's talk a little bit about that. So this idea of negativity can influence even passive transport of cations in the cell and anions out of the cell. Okay, remember cations being positive will pass into, of course, attraction and then repulsion with anions out of the cell. So whenever you're really talking about movement of particles and diffusion, we need to add a little bit more complexity. And there's two forces that drive the diffusion of ions. And this is more particular for ions, right, across the membrane. You have a chemical force, which is the ion concentration gradient, and you have an electrical force, right, which is the effect of the membrane potential on the ion movement. Sorry, my apologies. Okay. So these two forces, you have a chemical force and an electrical force, and combined together to create an electrochemical gradient. Okay. Okay, an example is sodium plus ions that are inside a resting nerve. There's generally low concentration inside the cell. When a cell is stimulated, gated channels open. Okay, remember you have sodium and outside the cell and inside the cell. And what could happen is that sodium ions can actually go and diffuse inside of the cell just because of a concentration gradient. Okay, so concentration gradients and electrical chemical gradient can work together because it's negative inside in general. Sodium is a positive ion. It would like to go inward into the cell. Okay, normal diffusion, both electrical chemical gradients and just concentration gradients in general work together. But that doesn't necessarily mean that that's always the case. It could happen where you have to go in opposite directions. And here's where the idea of the sodium potassium pumps come into play, right? And focusing more now on this particular panel. So here you have a specific example, a sodium potassium pump, active transport. So this pump, and let me just fix the typing here, it translocates 3 sodium plus ions out of the cell, okay, for every 2 potassium pumps into the cell. Whenever this occurs, there is a more net positive charge out of the cell, okay, than inside of the cell. So this electrical difference will create more energy, will create more voltage. Okay, If it's more positive outside, then it becomes less positive inside. Therefore, it's more tends to be more negative in the inside of the cell. So, um, a transport protein that stores energy as voltage across a membrane, we refer to them as an electrogenic pump. And in this case, that sodium potassium pump in animal cells is an example of that, where you want to create a difference, okay, of energy, right, where you want to form voltage. It's a major pump, very, very important um, for signaling, okay. So going into more specifics, you do need to know this whole process here. Not too difficult. Let's just follow the panel there. So initially, what happens? Um, let's break down the diagrams, right? So you have your cell membrane here, your phospholipid bilayer. You have the extracellular fluid in blue. You have the cytoplasm, the inside of the cell, in orange, right? So initially, you have these concentrations. Sodium is low on the inside, but potassium is high. And on the extracellular fluid, you have sodium high concentrations and potassium low concentrations. So what happens is that the cytoplasmic sodium comes in and binds to this carrier protein. And you can bind 3 sodium ions, okay, in these slots of the protein. That will then trigger a phosphorylation of this particular protein. Essentially, ATP comes in and will provide a phosphate, will phosphorylate, okay, the carrier protein, and this will induce the carrier protein to change shape, enabling sodium to be released into the outside of the cell. So all the time you're throwing out 3 sodium ions, okay? The carrier protein changes shape. Then what happens is that potassium ions, okay, and as it's changing shape, I want you to notice here that the binding sites for sodium ions also changes shape. So you really can't, you don't get sodium to go in a reverse way because it alters their shape. But this activates a binding site for potassium, and 2 potassium ions come in. They bind in. This forces this phosphate group to detach, and as it detaches, as a consequence, the carrier protein will then revert to its original shape, and the potassium ions will be liberated inside of the cell, okay, in the cytoplasmic area. So what's happening? Ultimately, since you're having 3 coming out, 2 coming in, there's going to be a more positive area outside of the cell than inside of the cell, okay? And this in itself is the sodium-potassium pump, okay? Again, useful for creating a voltage potential, right? There's other types of pumps that we're going to take a look at. There's a proton pumps, and this more specifically will help transport hydrogen ions, okay, hydrogen plus ions, through the membrane. We're going to see this in much more detail with cell respiration and the electron transport chain. Ultimately, you want to create a concentration gradient with hydrogen to create ATP, okay, for ATP synthesis. So in itself, it's stored energy for cellular work, okay? Again, this requires energy. This requires ATP. Okay. Going to more detail now is a kind of combined form of the 2 transports. You have cotransport. Okay, so this is a coupled form of transportation of substances within membrane proteins. So a solute that exists in different concentrations across a membrane can do work, okay, as it moves across a membrane by diffusion down its own concentration gradient. Okay, so think about that for a bit. As a molecule, just by having a concentration gradient from high to low, as it's passing through a protein and down the membrane, that movement, that energy that is carried within the movement can help another molecule go and move also. Okay, so this is called cotransport. Analogous to water that is pumped uphill and performs work as it goes downhill. So this is called co-transportation or co-transport. And then you have your transport protein, which is the co-transporter. Okay. So ultimately what it does, again, as I mentioned right now, it can couple the downhill diffusion of a solute to the uphill transport of a second substance against its own gradient. So one diffuses and the other one can create, the second substance can create a concentration gradient. Okay. Plant cells can do this. They use a gradient of hydrogen plus of hydrogen ions to generate ATP powered, generated by ATP powered proton pumps. Okay. And then that can drive the active transport of several biological molecules like amino acids and sugars, nutrients in themselves. Okay. So let's take a look at one example in detail, and you should understand this. To be able to explain it. The hydrogen ion and sucrose transporter. So what happens here is that hydrogen returns into the cell. Okay, and you can see that here. So you have a concentration of hydrogen ions. They're being forced, or they're diffusing into the cell. Okay, let me just move down a little bit here. They diffuse into the cell, but they also help translocate sucrose against a gradient. Okay, only if done with hydrogen plus ion. So only if it's present. So you have this kind of complex protein here where hydrogen binds and it can be helped diffuse down, and at the same time sucrose can also bind, but it's creating a concentration gradient of sucrose. Okay, so it's going from a low to high, and the hydrogen ions are going from high to low. Okay, the co-transporter. So hydrogen ions, they use this, this way to diffuse down their electrochemical gradient, and then that gradient is formed from, in the vicinity you have a proton pump. Okay, where you have these hydrogen plus ions, they're being shot back out into a gradient by ATP. Okay, these proton pumps aren't necessarily for, I mentioned earlier, aren't necessary for photosynthesis and cell respiration per se, but they are present right on the cell membrane to create hydrogen plus ions concentration. Okay. So again, very, very important that you can work together. One active, one passive transport can be working in concert. Okay. So plants use this hydrogen plus ions. Plants use hydrogen ions and sucrose co-transport to essentially load sucrose in the veins of leaves, so they can be distributed into the vessels and then distributed throughout the whole body, the whole plant body. Animal cells, we have a similar thing with sodium ions into intestinal cells that work together with glucose, so we can get glucose into the blood vessels, into our body, so you can move glucose throughout the body. Okay, very important in terms of treatments for certain diseases, right? Like diarrhea. What happens? Okay, why is diarrhea necessary? Pretty bad, especially in back in the times when you didn't have these electrolytes substances to help regain sodium. and salts, electrolytes, essentially. To babies, there would be a high mortality rate. And what happens in diarrhea is that the sodium sodium ions in waste, right, would normally be reabsorbed in the large intestine, in the colon. But when a person that has diarrhea will release everything too fast for sodium ions to be absorbed. So what happens is that you have a loss of electrolytes, you have a loss of these ions, and then you can't regulate the body to absorb and transport glucose and process glucose. So patients would die from diarrhea. Okay. So what happens now, of course, in modern day, we have electrolyte solutions that can easily, little salt packets, Gatorade, for example, even you can restore your electrolytes so you can have your salts there to help with this Cotransport system. Okay. Yes, there used to be a time where there would be high infant mortality worldwide just because of having diarrhea, just because you couldn't maintain salt in your body. So it's quite interesting. Okay. And the last thing we're going to talk about is 7.5. Bulk transport across the plasma membrane occurs by exocytosis and endocytosis. So we've talked about small molecules like particles that the cell needs, ions, sugars, and so forth. But what about incredibly large molecules like proteins, polysaccharides, and bacteria even, right? And pretty large structures that also need to be transported either inside of the cell or released outside of the cell. The way that this happens is that you would move this material via vesicles. Okay, they're packaged in vesicles. So let's take a look at them. Starting first with exocytosis. So, proteins and large molecules, they could be liberated, released outside of the cell via these vesicles that would fuse with the plasma membrane. This is called exocytosis. Okay, so as you're seeing here, an example would be the Golgi. The Golgi is creating these vesicles and they're shipping, whatever substance you need, secretory proteins. And these vesicles inside the cell are being shipped off, right, onto the cell membrane. The vesicle fuses on the cell membrane, merges, since the vesicle itself is made up of membrane, it merges with the cell membrane and it liberates the contents to the outside, okay, for these secretory proteins. Now, at the contact points, there's very special proteins that would help rearrange the vesicles, the lipid molecules to merge with the membrane, ultimately releasing the content. Okay, how the vesicle would initially go to the cell membrane is by microtubules that we talked about with the structures of the cell. Okay. And then, of course, depending on the nature of the secretory protein, you can use it for let's say, signaling and whatnot. Okay. So, you see this behavior of exocytosis more on secretory cells that they need to liberate proteins, enzymes, and whatnot. Pancreas secrete insulin into the extracellular fluid, for example. Nerve cells secrete neurotransmitters from one neuron to another, from one neuron to muscles, sorry. Plants, they can secrete proteins and carbohydrates outside of the cell for any metabolic processes. Okay? So, exocytosis, right? Let's move on to endocytosis. There's different forms of endocytosis that we're going to talk about. So the cell takes in molecules and any matter, transporting it within a vesicle that it will be formed into the cell and ship it wherever it needs to go within the cell. So it starts from the cell membrane, moving inwards. Endo meaning in. Okay. So it generally does look like a reverse of exocytosis, but there's different proteins that are involved in terms of like contact and contact of the particle that you want to go inside, and then the subsequent processes. Okay. But in a nutshell, you have a reverse. The membrane sinks inwards, creating a little depression. You get this formation of pseudopodia. There's like an extension of the membrane that will begin to cover any content that you want to build in, that you want to bring in. Okay. Essentially creating a vesicle. That pocket enlarges, pinches inward, detaches from the cell membrane, ultimately creating a vesicle, right? So there's three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. Okay, if we take a look here. Sorry for the quality of the image isn't so great. So let's say you have a large food particle you want to bring inside, referred to as phagocytosis. So this process produces little temporary extensions of the cell membrane. They're often called pseudopodia. It essentially wraps around the food particle, as you can see here, and brings it inside the cell, often called a food vacuole. Okay? And then if you need to digest it, it can fuse with a lysosome containing hydrolytic enzymes. Okay, here you can see an amoeba. An amoeba is starting to create this pseudopodium that will engulf essentially a algal cell. Okay. Phagocytosis. The next is pinocytosis, where you're getting a large number of small particles, solutes that you need inside the cell relatively quickly. Okay. These vesicles can have this pit is essentially referring to the depression, what is going to be the vesicle, but it could be surrounded by coat proteins. Okay, and these coat proteins, sorry, let me take this little symbol out. Okay, and these coat proteins can help enable the vesicle go to its target location a lot better. Okay, coat proteins. Doesn't necessarily have to have. Okay. So, in many cases, the parts of the plasma membrane that form vesicles are lined in their cytoplasmic side by a fuzzy layer of coprotein. Okay, these pits and the resulting vesicles are called coated pits. Okay, but it just helps for direction. Okay, and then you have receptor-mediated endocytosis. So here, again, you have a similar process of endocytosis, but the vesicles that are being formed here have special receptors, okay, very specific to a particular, just one particular type of solute that you want to bring in. Okay, just okay. So receptor-mediated endocytosis, very, very specialized. When you're dealing with receptors, you know you're going to get something very specific. Okay? It enables the cell to acquire bulk quantities of something specific. Okay? Even though you don't necessarily have a lot of it in the extracellular environment, just by having a receptor, you can create a good concentration of it. Okay? So specific solutes bind to the receptors. The receptor proteins then cluster in. They fold inward. Okay, so you can see here, they're folding inward. And once you have a good amount of the volume that you need, and you're creating the vesicle, ultimately, it'll pinch inward. Okay, receptor proteins then cluster into these coated pits, and each pit forms a vesicle containing these bound molecules. So this ensures, it's a way of insurance to bring this particular thing inside of the cell. Okay. After ingested material is liberated from the vesicle, the emptied receptors are recycled and then they'll be placed back into the outside of the cell. Outside of the cell. So they could be reused again. Okay. You have your little coat proteins, you have your plasma membrane, and here you have an example of a coated pit ready to be sent inward from the cell. Okay, so you have three forms of endocytosis. Okay, so in this case for humans, right, and cholesterol related endocytosis. So generally, what happens is that you'll take in cholesterol from for membrane synthesis. So cholesterol is very very important, right, and for the synthesis of other steroids. It's a good kind of base molecule to create other steroids. So cholesterol travels in blood within the blood, and particles that we have probably heard of called LDLs, low density lipoproteins, which is a complex of lipids and proteins. And so these low density lipoproteins would bind to receptors on the plasma membrane and then enter by endocytosis. Okay. This is what naturally occurs. But you could have, some patients have inherited diseases called hypercholesterolemia. And this is a condition where you have high levels of cholesterol in the blood just because these LDLs, they cannot enter the cells because the receptor mediated endocytosis, those receptors in particular are defective or they could be absent. So if you can't have these receptors, how are you going to collect the cholesterol, right? So, and these proteins, right? So these LDLs accumulate in the blood, and this could lead to atherosclerosis. You have lipids building up in the blood vessels. This could lead to a lot of lipid-related diseases like heart damage and even a stroke, right? So this is a kind of a medical related in terms of um, endocytosis, and this is actually a receptor mediated endocytosis. Okay. All right. So with that being said, I think we have finished chapter 7, and this also finishes chapter 2. Hopefully covering the content of of what is needed for the unit 2. For unit 2. All right. Hopefully this was helpful, guys, and I'll see you in another video. Thank you for watching.